Hello and welcome to the third topic of the cell biology subtopic of IB biology. And within this topic of membrane structure, I'll of course be talking about the membrane structures and the certain objects and molecules that are involved in the structure. Then I'll be showing you the fluid mosaic model and lastly talk about freeze fracture. Hope you enjoy. So first of all, the cell membrane is composed of a lipid bilayer with proteins moving within that said bilayer, and it provides a shape for the cell, but it also encloses its contents, so it acts as a form of stability for the cell. And the plasma membrane is in contact with the extracellular space. Additionally, the accepted model is the aforementioned fluid mosaic model, although there are different models that are also used, but are not as current in science as they are supposed to be. So moving on, we start with the phospholipid bilayer, and you can see a graph or an image of it on the left hand side, where you can also see the phosphomolecules with a hydrophilic head and a hydrophobic tail, and the head here actually consists of glycerin phosphate, as you can see here, and the hydrophobic tail consists of saturated fatty acids and unsaturated fatty acids, forming this fatty acid tail. And because it's composed of a hydrophilic and a hydrophobic layer or structure, it's called an amphipathic molecule, so it's both hydrophobic and hydrophilic. And basically it provides a transition between the intracellular and the extracellular space, as you can see here, and the fatty acid tails are showing towards each other, whereas the hydrophilic heads show on the outside. And the properties of said phosphomolecules are that they're held together by a weak hydrophobic interaction by the tails, as you can see here. And there's a layer, it provides a layer that restricts passage of substances, so it controls the permeability. Furthermore, these individual phospholipids move, and this provides membrane fluidity or membrane flexibility, and that so-called fluidity also leads to spontaneous breaking and reforming of membranes, called endo and exocytosis, or allowing for endo and exocytosis. Embedded in that phospholipid bilayer are membrane proteins, and membrane proteins are basically amino acids localized according to polarity, and there are also nonpolar amino acids that are nonpolar hydrophobic amino acids that associate directly with the lipid bilayer. And the polar amino acids are located internally, so if you can imagine a structure of a channel protein, for example, it will be nonpolar or hydrophobic on the outside, where it actually comes into contact with the lipid bilayer, whereas it will be polar or hydrophilic on the inside, where it does not directly contact the lipid bilayer. And there are two kinds of membrane proteins. On one hand side, we have integral proteins, integral membrane proteins that are permanently attached to the membrane, and those are often transmembrane proteins. And the main transmembrane structures are helices and beta barrels, so-called tertiary structures. And examples that you can see on the left-hand side, in one you see an alpha helix that's there for recognition and receptors. You also see into a helical bundle that's there for enzymes, transporters, and receptors. And then in the third picture for the integral proteins, you have a beta barrel that's also a transporter or channel protein. So you see the this variety of transmembrane structures. But then you also have peripheral proteins that are only temporarily attached by non-covalent interactions, and they associate with only one surface of the membrane. And of these peripheral proteins are most often enzymes and are therefore anchorage, transport, or so-called carriers, as you can see in the examples shown in 1, 2 through 3. And I haven't written down the exact structures for those, but as I already said, they're mostly therefore carriers, whereas those of the integral sort are transmembrane proteins most of the time. And there are also so-called so-called functions of the membrane. First of all, we have it functions as a junction, so it connects to cells, so-called intercellular joinings. We have the fact that it's enzymes, so it's therefore enzyme activity, and it fixes to membranes to fix metabolic pathways for these so-called membrane proteins, or the membrane proteins. Then we also have the transport as a function of membrane proteins because of the facilitated diffusion and the active transport. There's the recognition that functions as markers for cellular identification or cell-to-cell recognition. There's also a process called anchorage that's a function of membrane proteins. That's, as I already said, for anchorage and attachment, and it provides attachment points for the cytoskeleton and extracellular matrix. Lastly, we have transduction that functions as receptors for peptide hormones. So as you can see, membrane proteins have various different functions in and of themselves, and are therefore integral for the cell membrane. And for the last part of the building blocks of the cell membrane, we have cholesterol. And cholesterol is a component of animal cell membranes, and it's there to maintain the integrity and mechanical stability. And here, right down here, the hydroxyl group is actually hydrophilic, whereas the other part of the molecule, this entire rest, is hydrophobic. So it's also to some extent amphipathic. Moreover, in plant cells, plasma membranes surround and are supported by the rigid cell wall made of cellulose, which you should remember from the prior video. And the aforementioned hydrophilic part of cholesterol aligns this part here, the hydrophilic one, aligns towards the phosphate heads of the phospholipids, and the rest actually associates with the phospholipid tails, so it becomes part of the structure. And cholesterol interacts with the fatty acid tails to moderate membrane functions. For example, it immobilizes the outer surface of the membrane, therefore reducing the fluidity. It makes the membrane less permeable, and because it separates the phospholipid tails as they interact with them to prevent crystallization. And furthermore, it secures peripheral proteins by anchorage. Yes, moving on, we have the fluid mosaic model that I mentioned right at the start. And there are the phospholipid molecules I talked about, peripheral protein A, an example of a peripheral protein. And another thing I haven't talked about, namely the glycoprotein. And the glycoprotein is basically a protein plus a carbohydrate that's there for membrane stability, cell-to-cell recognition, cell signals, and receptors. This glycoprotein is again shown in the phosphate head and the fatty acid tails. And here's the cholesterol molecule I talked about, and these are two kinds of proteins, transmembrane proteins, integral proteins, and I'll be talking about the membrane transport in the next video, so I won't be going into that. But in general, when we talk about channel proteins, they go through the hydrophobic interior to enable water-soluble molecules to pass through with the help of facilitated diffusion. Another thing that I don't think is in this structure, in this graph here, are glycolipids that are attached to the phosphate heads sometimes, and they're also there for cell signaling, cell recognition, and stabilization. Here we also see a 3D structure, so you see the extent of the membrane, the membrane and the membrane proteins, and the way it is built when you look at it from a 3D structure, or at least an excerpt of said membrane. Lastly, we come on to freeze fracture, which is also called freeze etching sometimes, and it's a technique for preparing and viewing cellular membranes with the help of electron transport. And it's basically the freezing of a cell and fracturing it, as the name suggests, to make the inner surface of the membrane be seen through the electron microscope, as you can see in the picture on the side. There's the technique of separating the membrane into the extracellular layer and the cytoplasmic layer. And the technique is as follows. Well, the intermolecular bonds are split apart with the fracture. That's happening by immersing the cells in a chemicals that alter the strength of the internal and external plasma membrane, and thus immobilizes the macronutrients. The cells then pass through glycerol solutions to protect cell from bursting throughout the fracturing. And lastly, the cells are fractured in so-called helium-vented vacuums. Then they are shadowed with heavy metals and stabilized, so they are seen. And lastly, they're replicated. Their 3D structures replicated and viewed under the electron microscope, which is also how the fluid Mosaic model was actually first discovered, I believe. So yes, this was the third topic of membrane structures. These are my references, and I thank you for listening.